So today we are going to be taking some notes on HLA matching. So HLA is an acronym that stands for Human Leukocyte Antigen. And the way that these works is you have your body cells, so your skin cells, your nerve cells, your muscle cells, and on the surface of these cells, there are these proteins, these antigens, and they have different shapes. So we're going to just draw a whole bunch of different shapes on these. And I'm using different colors just because color is an easy way to visualize something. So these things that we just drew, these are the HLAs. And what they really are proteins on the surface of human cells. Now, other organisms also have leukocyte antigens, but they're not human leukocyte antigens. So dogs have antigens on the surface of their cells, horses, cows, all the different animals have these proteins on the surface of their cells, but the human ones are the ones that we have. So this is an example of a body cell. Now, body cells interact with immune system cells. So your immune system cells have these receptors on them. This is an immune system cell. For example, this could be like a T cell. And so the way that these work is that immune system cells use the HLAs to determine if the cell belongs in your body. So if it recognizes that as part of your body, then it will be fine. But if the immune system cell recognizes it as foreign, or if it does not recognize it, then they will destroy it. So if immune system cells don't recognize the HLAs, on a cell, then they will destroy it. What this means for people getting donations, either of tissues like blood or of organs like a liver or a lung or a heart, is that the donor cells the donor cell HLAs must match recipient cell HLAs. To avoid two things. The first thing that they're going to try and avoid is rejection. And rejection occurs when the host immune system or the recipient immune system attacks the transplant. And the other thing that we're trying to avoid is something called graft versus host disease. And in graft versus host disease, the graft is the donated immune system. And the donated immune system is coming from the fact that you're donating the stem cells that make the blood, and if the immune system cells come from the blood stem cells, so your white blood cells are your immune system cells. So when you donate a stem cell that ends up making a white blood cell, it's making an immune system cell. And those immune system cells belong to the donor, the graft. And so the donor has these immune system cells that they recognize the donor's cells as being good. But that means that if they don't match, then they recognize the host cells as being bad. And as a result, those donated immune system cells are going to attack the recipient. Now, graft versus host disease is going to cause a lot of problems because these donated immune system cells, if they don't recognize the host's HLAs, then what that means is that every cell in the recipient's body is open to attack because all of those host cells are going to have those HLAs that the immune system is going to recognize as bad, and it's going to try and destroy them. So how do we get our HLAs? Well, inside of all of your cells are your genes or your DNA, and your DNA is found on chromosomes. And this is a little diagram of chromosome 6. So chromosome 6 is where your HLAs, your genes for your HLAs are found. And so all along this chromosome, you've got these different sections that are genes, and they code for different proteins. And the genes for your HLA proteins they're found near the top. And if we zoom out on this, then what we have is we have this long section because we've zoomed out, and we can see that there are even more genes in this section. So you've got a gene for what's called HLA-A, and then you've got two genes kind of close to each other, and that's going to be for HLA-C and HLA-B. And these are named in the order they were discovered, not in the order that they're actually on the chromosome. And then at the bottom, you've got three more. So you've got HLA-DR, HLA-DQ, and HLA-DP. HLA proteins are coded for by genes. And you have two chromosomes, two copies of each chromosome. So you have one from mom, and one version from your dad. Which means that you have two versions of each HLA gene. So when we say we're trying to find somebody a match, what we're looking for most of the time is we're looking to have an 8 out of 8 match. And what that means is for HLA-A, C, B, and DR, both genes, so both proteins for each HLA are a match between the donor and the recipient. So you might have HLA-A 26 and HLA-A 112, and if both of those match, then you would have a 2 out of 2 match. When you then look at HLA-C, maybe you have HLA-C 46 and HLA-C 7, and if both of those match and both of the A's match, then you would have a 4 out of 4 match. So as you can see, there are 4 here. There are 4 genes, and if you have 2 versions of each, that means you have 8 different HLA proteins that they're looking for. But you have 6 different HLA proteins that are coded for on chromosome 6. And it turns out that these two just aren't as involved in immune response, and so it's not as important that you find a match for those. Now, you actually have even more leukocyte antigen proteins. So you have even more of these HLAs that you could match, but as the minor ones are not nearly as involved in HLA matching and rejection, we don't really worry about those, even though there are a lot more of these genes on the chromosome. So when we take a look at what we're trying to match here, we're trying to match these proteins, which are antigens, with the receptors that your immune system cells have. And because your immune system cells are able to identify your body as belonging, they're going to always recognize the HLAs for your body. Now, this is similar to blood typing, but because there are so many more versions of these HLA proteins, blood matching is actually a lot easier to do. Like the options for blood types are A, B, AB, and O, and you can be a positive or a negative, but that's only like 8 different options. Whereas with these, you have 8 different proteins, and each protein could have over 100 different options. So you're really trying to match a lot more different things, which means matching is a lot harder. Okay, that's all for now on HLA matching.